Kaushik, you can start.
Well, can I start, sir?
Yes, yes.
Okay. A very good afternoon to one and all present with us today. Myself, Kaushik Nayan Sharma, Assistant Professor, Faculty of Pharmaceutical Science, Assam Downtown University, would like to invite you all on behalf of our department to this faculty development program in paradigm shift in pharmaceutical education and research in post-pandemic era. Today, our chief guest, the speaker, has already joined with us, is Dr. Pranabesh Chattopadhyay. So I would like to give a brief outline about his. Uh, he's currently working as a scientist at and head pharmaceutical technology division, Defense Research Laboratory, Tezpur, Assam. He received his PhD from Birla Institute of Technology and Sciences, Birla Institute of Technology and Sciences, Pilani. He had been conferred with prestigious Fellow National Academy of Science in 2019. He had 10 patents and more than 220 publications in international and national journal. 10 monographs and 5 book chapters he have already published. He also guided 19 PhD students, 3 postdoctoral students, and more than 30 MPharm trainees. He is a lifetime member of CPSCA. So I would like to welcome him to the session. So, sir, please, you can share your slide and you can please give his presentation, sir. And his topic is preclinical toxicity studies overview. All the participants, you have to wait for some time because as our sorry, all the participants, you have to wait for some time because they are having some technical issue, and we'll solve it shortly. I will come back to you. Just wait for some time. We'll back to you. Hello, sir.
Sir, very good afternoon. Sorry, I'm late at times. Actually, the unfortunately today is getting this your communication is somewhat difficult. Just give me 1 minute. We have already connected.
Okay, sir. Sure. So very good afternoon to all. Actually, I'm very sorry that I regret that I could not attend in time. As actually, you are getting my voice? Yes, sir. Yes, sir.
Actually, I am in outside in from my stations. I am actually IIT. That is another reason that I got not get the connected in well the time because the network issues is there. So anyway, so we not lose any times. I'm thankful to the all of the organizers. They have given me opportunity to speak some word about this one, about this particular topic, preclinical toxicology studies and the drug assay, a outlook basically. Preclinical testing is known as a laboratory test or new drugs for a new medical devices usually done on the animal subject to see if the hope for the treatment really works or it is safe or it can be go to the further to the human being. Basically, this is we are look for this particular in the preclinical studies. Basically. Any discovery process, there is we can divide it in the three stages. One is called the pre-lead. It is sometimes known as a discovery stage. Next is the IND, basically investigation and new drug. And third one is for the NDA, that is your already a new drug or application. As per when the IND is actually successful, then actually NDA should be filed, or NDA can be claimed that the drug is discovered, or it is called sometimes the biological letter of application. So if you get divided in the process, there is a two phase. One is called the discovery phase, and second was the development phase. So discovery phase up to the IND actually take this.
Sorry, sir, interrupt you.
Tell me, sir.
Yeah, Mr. P. Sudhir Kumar, kindly mute your mic. Mr. P. Sudhir Kumar, kindly mute your mic. Otherwise, I will remove from the group.
Yes, actually, many background noise is coming, and that is the
Yes,
biggest
sir,
issue.
that's right.
Anyway, we are getting the many artifact actually after giving the deliver the lectures. I think many people are using this mic actually. Anyway, now you can divide in the pre-lead, IND and NDA of the BLA applications. You can think that first one is a discovery, second is a development. And in between this pre-lead to the IND development, the toxicology studies in the animals generally done. And after this toxicology, we are getting the perform generally phase, that is your phase 3, actually last phase, but we are getting the phase 1, phase 2, and as a phase 3. This is called of the clinical trial parameters. So in vitro or the animal models, basically you can think there is many things you have to think how to go for the development. First one is the hypothesis of the testings. Whatever drugs we would like to develop it, how will be testings actually? And we have to make the hypothesis. What is actually, whatever the lead molecules you get it, what in the your probable receptor, what is what you can bind. And second is the probable mechanism of actions. If you do not know the mechanism actions, naturally we could not develop the animal model because many things animal model actually give this actually first idea that drugs is acting or not. Third is the parameters. This is a major concern of this one. This molecule may be active sometimes. Safety is many times for the safety reasons many molecules. It is all known to you. And that is in development the surrogated marker. The development surrogated markers is useful for when getting any phytopharmaceutical or any herbal drugs basically. Surrogate markers is always required to maintaining the quality of this particular molecules or whatever in the animal study, whatever your marking of the particular marker where you think that this drugs is working or not, actually this molecule is working or not. Obviously last one is the ADME and PK, that is your absorption, distribution, metabolism, excretions, and naturally the pharmacokinetic study is a prime concern. But potentials for effect, toxicity profile, dose regimens, route of administrations, all should be take the considerations in cases of in vitro and the animals models. Now, in coming the dose selection, the species selection is most important. The species how they select basically it kind of mimic to the metabolite profiles and the pharmacology of the particular selections. Sometimes when you go to the dermal cases, dermal toxicity or dermal sensitizations, guinea pig is ideal model. If you go to the cancer model, naturally the knockout mice or otherwise, you can take this the mimic to the metabolite technique. Even you are taking any neuropsychological disorder like your Huntington disease, like Alzheimer's, there is many models you can do it, and by getting the particular deleting of the gene actually, we get the model actually, though it is expensive matter. Veckels is another this one issues that a solution versus suspensions. Sometimes many toxicity report has come that it is non-toxic, but ideally when the molecule is soluble or not, or in a suspension form, you should consider. Then toxicity you can take it. And concentration is another this one, and towards the vehicle concentration is there. Sometimes the 5% CMC and 10% CMC, you get a different type of release pattern. So the all the pharmaceuticals is there. Methods, how the formulation is takes and how the formulations may be made, and because sometimes a freeze dryer material, amorphous material, you get the somewhat different activity than crystalline. So even drug behaviors and amorphous and crystallines are some differences is there. Formulation, bioanalytical and immunological, especially the biopharmaceutical, generally are take the consideration in particular issues. And naturally, last one is a clinical plan. How we'll do the clinical trial, but my limitations is only the preclinicals today, and I'm going this one. Now, species selections, I have told you that is one of the most important parameters. If you go to the species, the dog is much more, that is a predictive versus non-predictive. Predictive means whatever you predicted, and that's maybe the gives good result actually. Your maybe the predictive means your whatever thing that is maybe a good. In most cases, the ideal predictive is a dog. Dog means the Brigel dog basically. The Brigel generally used in these cases. And primates is the second choice in these cases. For the dumping studies, dose studies, the primates is used. Primates basically McCoy is required in these cases. McCoy is ideal for primate studies. The rat has a very good relationship between the predictive and non-predictive. If you get the predictives also mean predictive mean what I say, that is suppose you are taking the Huntington disease models, a rat could be used, but sometimes it's a non-predictive. Because the normal rat could not be given the response of the Huntington disease. There's some genetic deletion is required particularly, and also some Alzheimer's models. Also, people are looking, by amyloid formations, and they are getting the Marvel test or like the bullet states. But but ideally, this is not the model. You get these models, you will get the same mimic non-specific as. Mice generally is not used for any for studies of the efficacy studies basically. Mice is a non-predictive is much more. Even the people prefer the mice because the molecules is very small, dose required very small, but mice is ideally the people prefer. But with the species selections of the particular in predictive one, your response, mice is a less considerations. Next one, now disease pattern. Many things could not be possible to determine in the animal model, like cramps, sweating, dry mouth, headache, and dizziness, nausea, is not possible in any animal model. It is only the humans is a prime consideration. But actual animal models comes from the skin toxicity. The skin is somewhere the relation between the skin is there. Then you can go through that is the skin, hypertension, blood sugar, gastrointestinal disorder, vomiting, alopecia, tremor, salivations, humoral responses, any hormonal responses. CNS studies, myocardium, yes, animals is giving the very good response in these cases. Even up to the ataxia, if you go to the convulsions and ataxia, human could not be used. And that's why we are taking animals these cases. So you can understand the limitations of the animal studies. I told you that crimping and sweating could not be done. Dry mouth study could not be done in animals. Whereas in ataxia and in convulsions, studies, reproductive and toxicity studies, some of the animals is restricted. We will not get much more information in these cases. So now in the process of the preclinical trial, any ideal pharmacology, if you think we have followed mainly the 5 steps. First one from the YOLO actually start the drug target and interactions. It is the one of the prime considerations. Second step, development of the bioassay. Third one, the screen the drug in the assay. Fourth one, the establish of effective and toxic doses, and fifth one, the file for the approval on investigation new drug. Yes, well, we go to the stepwise. First, we'll go to the drug target and the interactions. So, I am showing one, one video to you. I think it will be shareable to you all, and I think sharing to you all. I think you will get this video. Just go to the Alzheimer's model one. We are getting from the internet actually. This is known as a buried model basically, and this is a mimic of the Alzheimer's model. Alzheimer's is one of the devastating cases. That all the amyloid formations is there, and people could not memory, could have their efficacy of the human behavior is less. So they sometimes forget actually. Yes, it is the drug development. It is very difficult in these cases of Alzheimer's. In these cases, the marble models is one of the good models actually. If you go to the amyloid formations, generally these mice actually they forget actually where the marble is there. If you put the marble in the animals see that it is the behavior of the animals. The Alzheimer's generally animals could not get it. They are not buried actually. They're simple. There is a very lazy types of their behavior. But it is a normal mice basically. See that if you put the buried, you will even experiment also you will do. If you hide the buried, they are buried and they will take this marble basically. It is a marble test. Just see the slide. Oh, sorry, okay. So virtually, this marble tested, that is Alzheimer's, how this active and how much is passive, basically response of the animals, and due to the lackness of the memory. And in build, the rat has the property that rat and mice both have the rodent has the property to digging out. But Alzheimer's, when they produce in the rat or mice, they forget to the digging out. That's why it is somewhat difficult. And symptoms, you know, that Alzheimer's is the most common types of dementia, actually. This is called the dementia. And because due to the dementia, they could not get this response, basically. So we'll not take the much more of this particular slide. This is this acknowledgement to the Cambridge University. They have this particular video they have uploaded. And see, this is, thank you so much to the audience that we were giving this one. Next, actually, you know, receptor interaction is one of the biggest challenges between the target. What is the target of the drugs and how this act actually, either cellular or genetically, or the belief to the association of the particular disease. You know, many types of interactions is there, and competitive agonists and your allosteric agonist, antagonists, both are there. But it is helpful to the treatment of the particular specific disease, you know, the particular receptor. I'm giving one example in the respiratory systems where we work basically. Adrenergic agonist COPD, salmeterol and xenophoid is one of the very good this one. Even the leukotriene antagonist, montelukast, could be used commonly used for in the respiratory systems. So how this mechanism takes place, you know, the leukotriene formations and leukotriene, how this stabilize the mast cell, the montelukast, the people such knows. Basically, the receptors, they are highly binding of the montelukast with the leukotriene receptors, and we will get the efficacy in these cases. Receptor-like interactions, one of the giving the example category. Sometimes receptor and interactions might be the very, very fatal sometimes. Beta agonists like the salbutamol, it is normally using all the asthma in the cases, but generally the 0.3 milligrams in meter dose they are using, in per puff, per doses. But if you get the higher of the doses, the people that try for the 2 or 3 puffs, immediately you get your heart palpitations and residual because residual presence of excessive of the beta receptor antagonist. It is a bit, but partially your salbutamol in the high concentrations, it is not limiting to the beta receptors because it is a non-selective. They are good to the beta 1 also. Beta 1 is giving the heart actually and getting the very big palpitation in cases. So that is the thing. And second is a reactive metabolite. We are also consider how the reactive formations spaces and basically toxicity is generally form actually when the reactive metabolism takes place. You know that the cytochrome P450 role, cytochrome, another cytochrome derivative, cytochrome P4, many of your isomerisms were observed. According to the isomerisms, the cytochrome generally affect actually. And in the functional group, the generating of the metabolites is one of the biggest challenge, and their hepatotoxicities are very common. And genotoxicity is observed in DNA in this case as the most common target of mechanisms. So xenobiotic, non-xenobiotic metabolisms, it could be eliminations, reactive metabolites, binding to the cellular molecule, and generally it to the enzymes, receptor, and metabolism of the DNA. Then toxicity is produced, and tissue injury and cancers in the case of cellular repair also in these cases. So reactive metabolism is one of the biggest thing. Many reactive metabolisms there, like Roxabrine, and one of the major concern of this one, and this is a basically drug model. But if you get the P540 isomerism, A2 isomerisms, you'll get the chlorine generally induced. In these cases, they will easily can bind in the protein, and this protein starts giving this very devastating effect. So tetrodotoxin is one of the particular groups, and this is one of the major toxicity you can observe in these cases. In common examples, you can get the paracetamol. Paracetamol, is a very a common pathway. Paracetamol in the normal phase, phase 1 oxidation that takes place, and N-acetyl and benzopyrene in imine form actually takes place, and glutathione formations is there in phase 2 metabolism basically. Phase 2 they are combined with the glutathione and urine excretion takes place. But sometimes it is a reaction with the protein, and reaction protein if takes place in the N-acetyl and benzopyrene imine, naturally highly toxic will be observed in toxicity cases, and sometimes death could be observed in these cases. So paracetamol metabolism is another biggest issues. In sometimes the paracetamol give mainly for the hepatotoxicity. Development validations of the bioassay, and I have told you the preliminary planning and the decisions points have to take, and particularly how the development in the bioassay will do. Define the analytical methods of validation protocol. What is analytical protocol you have to take? What you would like to estimate, and what is the analytical tools in this case? So you have to first also validate it. Design methods in the validations is mind. Which design experiment you are doing, especially in the statistical analysis. According to statistical analysis, interpreting the group, in between the group, and you can take this one. ICST topic Q6B basically specification you have to follow in these cases of the you have developed the validations of the bioassay. The test procedures and acceptance criteria of the biotechnological or biological products and the ICSQC you have to here. Zebrafish embryo sometimes used in the lab for the drug in the bone density studies, blood vessels formations growth, even CAMS will do in the labs actually, and many other systems in the zebrafish. Anti-COVID testing drugs, in the animal model, really challenging. There is not establishing the anti-COVID drugs testings, and how this anti-COVID drugs could be established in the animal model. It is a big challenge. We cannot even people are trying, we are not getting the mimic models, and many bioassays is very difficult and challenging. So another clip I saw, it is very interesting clip, and because the two, that is also we'll get from the same internet, and two basically, McCoy basically, how the intelligent McCoy can observe. One McCoy is giving to the grapes, another McCoy giving the cucumber. But a second McCoy is telling why you're giving to the grapes to another one, why not to me, and how they are behaving. See that. Now he is not ready to give his particular the cucumber, and he is throwing out. No, I would like to take it only the grapes, like other McCoy, but he is telling, give me. No, he is not ready to get this one, and he is returning back. Well, I will not take it. See, he is also asking the same thing in the grade, and that is why we are telling, the animals model is the biggest challenge for how to develop it. Now, screening of the drug assay, the species selection is chosen in the bioassay, that is the particular species. Sometimes some species the receptors active, some species the receptor is not active. So we can take this species according to the receptors, and receptor abundance tissues are chosen for the bioassay. This will be well discriminated the drug safe or the effective in this case of bioassay. Species selections like non-adrenaline, the spinal cardinal, use pineal cardinal, using for non-adrenaline assay. Cardioglycosides, Dynapig is with a particular model. Estrogens, Ovatectusmide rat is a good model in these cases. Just give me one time. Okay. And estrogen cases, estrogen acid Ovatectusmide female rats is used. Isolated tissue sometimes used in common laboratory practice. Will do it. I think you are doing histamine assay. Gynapigilium is used. Adrenaline rat uterus is used. Oxytocin rat uterus. Estrogen is used. Isolated cells sometimes used for the plasma leukotriene hormone estimation by stimulation of testosterone. The isolated lytic cells generally used. And step 4, establish the doses is safe or toxic. Actually, the OECD or OECD 452 adopted 2018 generally used for the chronic toxicity for non-observed adverse reactions, what is called something NOEL dose, or it's a LOL, at the low observes your particularly effective level, or point to the departure of the establishment of the benchmark dose, which is called sometimes the BMD dose. So 455 generally used in these cases. LD50, ED50, and therapeutic index are the constant for any dose range study. If the therapeutic index is very small, then you can take the very precocious particular drugs like ganglion blocker, like many anesthetic agents. So TI should be taken very considerations. Observation in these cases: mortality, clinical observation, body weight, appetite. You have to observe in animals. Clinical pathology, serum chemistry, hematology, coagulation, urine analysis, pathology, target, non-target tissues, scheduled and unscheduled death. You have to also note it. And comprehensive gross pathology, organ weights, and histopathology you have to use. And pathology generally do the bind study in this case of treatment. So, preliminary range finding study, the maximum tolerated dose, MTT, dose limiting toxicity, DLT, up-down toxicity study, single and multiple dose ranging finding. All our methods is very good described in OECD, and according to OECD, you can do the range study, you can do it according to what your requirement. Subacute, subchronic, and chronic toxicity, generally single and multiple dose generally used, due to sense 2, 4, 13, 26. Same OECD has told this guideline, and you can go through it and do accordingly. Toxicology test battery, generally we are taking this many parameters. CNS study, the CNS function test, motor activity and behavioral changes. Cardiovascular studies, and in this case ECG and hemodynamic parameters. In the animals generally you do. Respiration function test, gestational mortality studies, hematological evaluations, blood coagulations, hepatic and renal function stakes, we can take it as a consideration. Now, step 5, applications for the made of IND. Now, why you are telling the IND? When you know the molecule, you are going to good in response in the animals models. Immediately you file for the patent, you take your IPR as urine for you and file the IND. Investigation new drug. Sometimes many discussions takes place. We are in the limitations. We are in researcher. We cannot do many industrial setup. We cannot do the many thing. We have the many limitations also funding. But I will prefer you go to the IND filing. IND filing is very easy actually. You can do yourselves also. After IND filing, you can ask the industry to come in and I have the molecules. I am ready to give you, and I have done IND studies. And this confidence, if you give the industry maybe come up to you, and maybe from your hand, maybe the new molecules may be arrived, and you see the essence that you are doing something. Now, we are taking the 4 parameters of the studies, like traditional, biopharmaceuticals, vaccines, and cancers. Single dose, yes, the DRF studies, yes, required, traditional cases. Biopharmaceuticals, 1 or 2 species. Vaccines cases, yes, 1 species is required. Single dose studies. Single dose studies means all toxicity will be done with single dose, like oral, acute, or your oral acute, you have to go to the your dermal acute, everything you done the single toxicity. Cancers also, yes, required, mandatory. Repeat dose toxicity is required in all the 3 cases, all the 4 cases. Genotoxicity is required only the traditional medicines, in these cases of I think the IND studies, and biopharmaceutical and vaccines not required. And yes, is in toxicity studies. In the safety pharmacology, in vaccines is not required, but in the ES, all the cases is required. Now, reformulated drugs or repurposed drug, actually, basically, again, the single dose can be done. Repeat toxicity, genotoxicity, safety pharmacology, almost the similar line. And basically, ethics and moral is a big issues I've taken from someone, and it is one of the big word. Chimpanzee generally not infected with the syphilis, but if you the previous studies, many McCoy studies, the syphilis generally injected in the animals, and then they go to the chimpanzee is using for the test bed. So this is not it could not benefit anyone. Only the response study, and this study could not be considered also. That's why primates is similar in many ways for the human, but they are also very different too. So if the primate studies or McCoy study, you will very consider in these cases. Impetus of the Nuremberg codes, that in this case, the Nazi issues, the Nazi physicians, basically Nuremberg, Germany, they have all the autocrats performed in the prisoners' wards, and they have the many unwanted chemicals and drugs actually used in the particular criminal, considering that is the war prisoners, but they are the war prisoners, they are not the criminals. So they have done the many things. There's a crime effect. Unknowingly using, in this case, Nazi basically is done in Nuremberg Code, and Nuremberg Code give a very good ethics actually. And Tuskegee syphilis experiment is one of the experiment in these cases. In the, I know it is called something bad blood study. If you go to the studies, you get the many things. How they have done the brutality is done to the animals model actually, and they have done the syphilis studies in the rather than animal to the human model, and it is published after the in 1993. The study has done in 1932 in these cases. So Tuskegee's Gagnetic White Cup actually, after this one, these two studies has embarked in the entire of the world. So now this human study is not, do the many procedures you have to adopted in these cases, institutional review, data review, and all the medical establishment you have to do it. So I'm giving the 2-3 examples in particular receptors and our working, and I'll be really, I am thankful to my students. Some students are in already a faculty in your institute, especially Manas is there, I think. Santa is there, Piali also there. They are all my students, and yes, I think some work we can show you. Even in these cases, we are going to the TRPV receptor basically. It is called the transiente receptor, the potential vanilloid receptor TRP1. It is basically heat sensations that takes place, and capsaicin activates it basically. You know the TRPV, many TRPV is there. That's the TRPV1, TRPV-MA, TRPV4, TRPV3, but TRPV1 will generate the heat actually, and this heat has this TRPV has actions in the respiratory systems as well as in ocular systems. So later on, we go to the many agents for the especially the using in incapacitating agents or sometimes the tear gases. In these cases, many chemical agents like atimetine, and generally adenosine is a DM category, an organic compound, technically an arsenal and dichronomine chloride system. Basically, their work also, giving the TRPV1 receptors, and it is called the vomiting and staging gases. CN is a chromphene-acetone. CN is known as the TR gases developed by the German military. Then compound 2-chlorobenzylene menonitrile. Common police also using CS and CN. I mean, if you go to the today's also, after this so long discovery, and there's all the military and all the police forces has used, though the US is not using these particular chemicals. And chlorobenzylene nitrite LCT around 35,800 milligram per minute MQ. In this case, we are not considering the LD actually. Any gaseous phases, any gaseous chemicals, we take the lethal concentration in respect of the time, how much 50 mortality. So it is known as LCT. So because if increasing the time transitions, naturally the people will be, it will be more toxicity will be observed. So in this case, the chlorobenzylene also there. It is using basically US defense, and dibenzoxipine also there. It is developed by the British military in the defense. And generally, we are working on the TRPV, and it is TRPV is more than 43-degree temperatures can generate, the heat can be generated. Heating generated, the sodium calcium channel basically is activated. And naturally, if we activate it, the other protein receptors also take the part. And the TRPV in 52-degree temperatures, the response, the temperatures, the response in the heat generation. And in this case, the TRPV more than 33. So many things, unexpected gas casualties. If you go to the Moscow, the medical toxicology perspective, in October 2002, the Russian military used a mysterious gas. This mysterious gas could not, they are revealed. In Chechen rebels is entire in the Moscow theaters, and later on it discovered erstwhile phenyl derivative. Serpentine and remifentanil are used because in using as an anesthetic, because they have thought that all the people will be the loss there means sensation. Later on, the police will be captured along with the particular the peoples with the Chechen military, the Chechen rebels. But unfortunately, they have not done the proper study, and LCT they have not determined, and all the people have died. More than 200 people have died, including the whole theaters, because this phenyl derivative is so potent, and the therapeutic index is so small, and everyone has died along with the rebels. So you can understand this is basically there. And now this is a TRPV1 actually. We are going on the particular in this case, the capsaicins actually act expresses. If you go to the slide, actually, acidic increase, heat generated, and ethanol can suppress. If you get it, if you go to home actually, even you're cooking with the chili, if you get an oil or some ethanol, you will get the relaxation. So we can't get the TRPV get the suppressed one. But in these cases, if TRPV is active in the presence of the vesicle in chili, basically, in the cases, phosphorylation takes place. The phosphorylation takes place naturally. The calcium and the tremendously increase in the inner side. So basically, this is a binding site that is a phosphorylation site, anchor protein, ATP binding, and calmodulin binding. So TRP domain basically is active in these cases. So we have done many things, and I told you that TRPV1 in the airway inflammations, and basically trachea and the larynx had the abundance of the TRPV, and in case of the intercellular calcium is an increase. So the myosin, C and C fiber to the myosin fiber, the vagus nerves is too much active in these cases, and giving the response of the inflammations, and mucinous gland cells, inflammatory bronchial constructs, and tracheal mucus edema, inflammatory cell, and chemotaxis will observe. So in these cases, in cases of the eyelid and TRPV M8 and cornea TRPV1 and TRPV1, this is in cornea positions where the TRPV abundance, it is a subtype of the TRPV, and conjugativity is a TRPV. So whatever the tear gases or any the chili when you do eye may come in the eye basically, the eyelid positions will be get the inflamed basically, in cornea get another inflamed, and conjugata is naturally the reddish you will observe. So in presence of the, even if you touch the the chili in your eye, or in presence of the tear gases or any gases, we are getting basically the chili gases in the eye. So basically our study is done based on the TRP vanillin receptors. I will give some slides, and and this is one of the study. We have done it in my students is there, and she is in NIPER. I think Dr. Harsita is there. See this is there is basically a rabbit eye in eye acute irritations. In particular, in these cases, you should take this normal albino rabbit could not be taken as a consideration. So if you do the inflammation studies in the normal your New Zealand or your albino New Zealand rabbit, the eye is already reddish. So it is very difficult to understand how much inflammation is there to identify in the visually. So in these cases, generally you get we are taking this one Juker rabbit basically. The eyeball is purely black actually. So you can observe that is the first slide that you one that is we are getting the in these cases it is a control rat actually control rabbit. The many macular formations is there, non-macular formations that exposes how many the macular formation is comes, And if you do the corneal fluorescence, this is basically changing basically in a rabbit. In this cases, this is the second photographs is getting to the capsaicins. So tremendous amount of macular formation is there because activation of the TRPV1. So you can take this one, all the studies in the inflammation studies also. When any fluorescence dye, if you're using in the eye, basically, if the your odema takes place, naturally leakage will be very high. So in these cases, you'll observe that number 2 is a capsaicin induced. Many leakages you'll observe. So how the inflammation takes place. Same as if you go to the fundus imaging, you will get by the fundus digital camera. In these cases, I have told that is a number 2 is in your control. In this case, not observe anything, and that is 1 and 2 is the control, and number 3 as 1, 2, 3. I think the control 1, 2 cases. Now see that is number 4 number. The 4 number is using the capsaicins, the activator of the capsaicins. So that is actually vein actually. That is your vein and how the inflammation takes place with the bluish color positions. And capsaicin is an antagonist of the capsaicin receptors. So basically the capsaicins you are using in these cases 5 and 5, 6, 7, 8, and you observe how much changes takes place. The inflammations and reducing of the vessels dilations is somewhat reducing. So compare with this 4th and 5th and 6th in these cases, we will observe this particular changing. If you get the retinal layers also, again, if you get to the third one, in this case is retinal layer, it is basically one is the retinal layers, and second one is the retinal pigment epithelium, third generally outer nuclear layer, 4th is outer calcium layer basically. In this cases also we observe that is how the capsaicins is taking the outburst of the pigmentations in this particular layers actually, and how this capsaicin take the antagonist particular receptors. So basically it is a OCT basically, occurrent coherent imaging. If you go to the much more inflammations, the diameter is increased. So I've told you in the cases of 3 actually, in these cases, if you do the second is your particular against the capsaicin. So I think this is on highest one. So this is a high much, maximum your corneal thickness is increased in presence of the activator of the capsaicin by TRPV1 receptor. But comparing to the capsaicin, that is some reduced, and this is same as in retina actually. The retinal that is in common chromatographs. In this cases, see the 441 versus 427. So 418 and 427, the thickness, it is using the capsaicin and capsaicin against and capsaicins. And capsaicin, I have told you, TRPV1 receptors. Then agonist, actually the antagonist, how this is reducing you can observe this one. So many things where can go, and many responsive techniques in these cases. And in cases although involving imaging actually, in cases also taking this imaging, when you develop some product actually, this receptors is only the eye and bronchus in cases. So we go to the imaging studies also. In these cases, not much more. It is limited towards only the respiration systems and your ocular systems. After the 240 minutes, actually, their release pattern, if you go to the release pattern, the capsaicin versus capsaicin somewhat reducing actually. But whatever the formulations we made, and that is there inside only the eye and mainly the ocular systems. So PG2 and PG2A is one of the this taking in these cases the marker actually. You know the activating acid pathway, you take it, that's the inflammatory marker. So PGE2 versus and PGF2 alpha, you can observe what is the changing, what is the concentration. So capsaicin increase highest, as well as capsaicin plus have antagonized the TRPV1 receptors activity. So TLR pathway, I'm really thankful to, I think the Pompey work actually. Pompey is shortly you'll get her PhD. So TLR7 also we are getting this. If you go to the TLR pathway, DAPI versus the TLR7's activations by TLR7's activated by the green actually. When in the March actually you will get the observe the TLR7 activity. So TLR7 will also express in these cases 10% of capsaicin. So it's a 10% concentrations effect. It's a DAPI. You get to the TLR7 highly active thing in these cases. When March it and see the expressions. So the control versus this one, it's somewhat is in higher range. So we are accordingly studies in many product actually developing, and almost 18 state of police is using this chili grenade actually, and it is one of the including 18 state police, and they are making it and selling. You are using their arsenal. This is all chili based basically. The one of the hottest chilies and the Bhujala case there in Assam. Fortunately, we got from there and ultimately developed it. So I'm not going to the many things. Just something I think many students also there, and I'll be end my lectures within few minutes. First, what we do not be much more go to the science. Fix the your requirement and target. Look at the deliverable. What delivery you like to do is my how we look out these particular projects, and when writing the project to any funding authority. So what is your requirement and what is the target you are looking? First thing. Second thing, never dream the irrational study, the raw material, lead compound, etc. Unrelated issues you should not think. So never dream in the irrational studies. So that is will give you the divert your pathway from your thinking. Third one, avoid extended studies. Know the steps, laws, and work according to the project allocation. So whatever project you like to do, and avoid the extended these types, and know the rules what said, and according to step you can follow it. Third one, pay attention to the correct route for the approval process, because what you are making today, world is not like your limitation studies. Though 2-3 papers you have made it and 2-3 work you have done. Today, all funding agencies are ready for either asking that particular what your deliverable. And deliverable means that should be the congenital delivery. So approval process is required. We are not avoiding the Drug Cosmetics Act. Even the many rules is there. Biomedical Devices Act also there. So we have to what is the approval process to take. So pay attention, choice your correct route in these cases. Fifth one, avoid repetitive research with the brand change. Don't do the repetitive studies. This today I have done, tomorrow this. No, you take this one molecules and go to the end actually, and avoid the different trade names in the same products. Avoid the survey. It is not the yield the deliverable. Don't go to the survey studies. Rather fix the problem from the old survey or other sources. So this is my one of the things.
And sixth one is a focus on your competitions. Who is with another people are there, what are they doing, and how we are, how can avoid to them in your study. Sixth one, listens what the consumer user says, what the other people are saying, what your guide is saying, what your other people are saying. Accordingly, you develop, frame the projects. And think the IPR protection. It will save you later rather. IPR, you have to take your. It is not give to anyone, because IPR is big issues, and you have to take you. Brand arrogance is uniqueness is there. Like Jandu, the great product for the pharmaceutical, phytopharmaceutical. Dabur have the own brand name, so you have to take the brand name and your uniqueness of the particular. I'm given two examples, interesting examples, rather than the science actually. Pepsi could not do this type. Pepsi have given this type consumer drinking for the breakfast times. It made no sense into the audience, because when the brand specific it was to be drink. Right product, because Pepsi is giving the energy, there are people that think the right product, right time, use and complete product is services to the industry partner. So take your industry partner to develop the product and throw in the market. Even the 20% share Kellogg's in India. Why? Kellogg's has big brand in the entire pan of world, but only the 18 million consumers is there. But Kellogg's is not people's are accepted. Why? What is wrong with the Kellogg's? Because we are eating the chapati. We are prefer this type breakfast. It's very possible in the UK and US, but we are there is no chance of the Kellogg's to the marketing. That's why Kellogg's is not introduced. So that what research you have to do, how this product could be made, and how it could be come out in the funding agencies. So this is the biggest issues. Simple, you making the nanoparticle and you seeing the cancer's activity, nobody would be fine. You are not nobody to fund you. So actual deliverable is required and uniqueness is required. So lessons that know your user, and your innovations required to users and not to you only, because you are people are looking to you. Innovations is looking to the entire pan of government actually, because they are giving to you money to you. There is no free lunch. So know that your market potentials. One of the big story in the Pond, suppose if you think the Ponds comes in the very good brand for the cosmetics products, but Ponds is make the toothpaste. Will you accept it? No, people are not accept, because Ponds has the toothpaste could not market, because it's a skin cream, skin care product. Every establishment has a brand value. So you can take this your research proposals or university that is your names, trade names, and that could be have the own brand value. I'm not going much more. That's my students. I think many juniors is there. Give the time to your precious time to realize the value of 1 year. Ask a student who has failed in his exams to realize the value of a 1 mark. Ask a mother who has given a birth to a premature baby, and to realize the value of a 1 week. Ask the weekly editor. And to realize the value of a 1 hour, ask the love strucks who are impatient to meet. To realize the value of a 1 minute, ask the passenger who miss the stream just. To realize the value of a 1 second, ask the persons who has survived an accident. And to realize the value of a 1 and 10 second, ask the athlete who missed the gold medal in the Olympics. So every time has a value. So you can make it accordingly, stepwise. You can make your project proposals, and that will help you. So second is vital 1/3 of the seconds. In Persian Gulf where the software glitch is there, responsible for the setting down. A prior mistakes was in the radar systems. So the software had become operated no longer in the recommended 100 hours instead of 14 hours. So they have using the 100 hours instead of 14 hours. The long operation, the radar timely 1/3 of the seconds. And result, this is caused the Patriot missiles to the misinterpret in the incoming Iraqi Scud that killed 28 soldiers and 197. So every times has own value, and you take it. Hidden dangers and look at that. Doctor saying don't go with a shortcut. And in Delhi, one prescription is written the injection CP. The nuts look at the CP to be she thought that is the chloroquine phosphate, but the doctor means CP is a chloramphenicol. There are many than 100 errors in the confusing Cerebrex COX-2 inhibitor with the Cerivaxin antiepileptic. The companies launched extensive education campaign to the doctors and everything. So yes, there is no shortcut. And validations, equipment, and qualification. Validations, equipment, qualifications. When incidents really is not happened, two blenders used in the production single tablet formulation, but you will get in both binder will form the same manufacturer, same model, but same design. Although one blender was older, another is the supplier told that drug manufacturer that the units were identical. But old blender not included in the process validations. In testing written the sample, the company found some lots of failed in the containing formatting specification. This is what traced in the old blender. That is, old blender had a different RMP set in the same settings. A result recalled 1.5 million bottles from in US in the foreign market. So many example you can give there. The counterfeiting one is the Karnataka told that is very interesting. Karnataka told the handover stamp scam kingpin. A counterfeit sold that counterfeited sold the fake stamps paper valued in 2,200 crore in over 8 years. Now Nasik security press sold entire printing machinery to a scrap dealer. The counterfeiter purchased the machinery and set up a security printing press in Bhiwadi. You know the counterfeiting that is stamp case. This one. What is the quality? Its quality is spending money does not promote the quality. Throwing the money at the problems does not mean the quality. Investing in the technologies on automation does not put quality. Quality is about attitude, and because we have required attitude. Many demand of the society, you can do the many drugs molecules. You'll ask future drug molecule, the DNA delivery, DNA modified drugs, gene product, genetics in the futures. Many things is there. They're asking, this is human kinetics in the trial. Where people are using this human as a guinea pig trial, like sometimes we are observing many things that many drugs is going to the unethical trial basically. Many journey actually starts. The life secret to the life DNA. We made the many lives, and many demand also there. Cancers. There is no answer still. We are looking for the new molecules. Even though last month we have observed America is claiming that molecules has came. This is all the big peoples, and they have millionaire actually. The richest lady is Kiran Majumdar. It is from the drug pharmaceutical. It is 2.5 billion. Dr. Deshbandhu 7.8. Lina Tiwari, madam, is 1.9 billion in USB. And the son is Dilip Sambisa. All are the big peoples, and they have mixed on the pharmaceutical sector. It is a great India, and we have to the reach in the cultural heritage also, and many plants will get, and you can get the developed. So great nations and great diversity is there. Acknowledgement news agency TV channel to group with my JRF students, Dr. Sita, Ms. Pompi, yes, Manas is always there, and other students also. So that is a slide designed by the site Carnival, slide designed by M. Jordan Smith. It's taken from and thank you so much. And through the innovation, transformation is a thought, is a technology. Thank you so much.
Thank you very much, sir, for your magnificent presentation and enlightening us with your knowledge and experience. Now, let's move on to the question and answer round.
Yes.
Sir, can you take the first question?
Yes, please. I can take the questions.
Okay, thank you. If anyone has any question, please ask. You can turn on your microphone or you can message in comment box. Anyone? If
I
there
think,
is,
right. Yes, Mr. Kaushik sir. I think people are not taking any questions. I think they have either learned everything, otherwise they have not understand what lectures. There's the two probabilities there. Anyway, thank you so much. Anything from your side, tell me.
Okay. If there is no question, then we can move forward. Now, I would like to request Dr. Durga Prasad Kemishetty sir, Associate Professor, Department of Pharmaceutical Science, Assam Datta University, to kindly present the vote of thanks. Over to you, sir.
Thank you, Kaushik.
Yes, sir.
Good afternoon, Honorable Chancellor Sir, Vice Chancellor Sir, Dean Sir, HOD Sir, and most valuable invited chief guest of today's session, that is Dr. P. Chattopadhyay Sir, and speakers of all the sessions. It is my privilege for me to give vote of thanks on this FDP session for this 6 days FDP. So almost we have come to an end of our 6 days FDP program on paradigm shift in pharmaceutical education and research in post-pandemic era. So firstly, I would like to say my vote of thanks to our distinguished chief guest of the day, Dr. P. Chattopadhyay Sir, Scientist, F. Defense Research Laboratory, DRDO, Tezpur, Assam. And one of the proudest thing for Sir is that the 3 PhD students of under Sir guidance are working with ADDU as a faculty. Thank you, sir, for sharing your valuable research experience on this platform. Then I would like to express my profound gratitude to our respected Dean Sir, Dr. Biplab Kumar Dey Sir, for his moral support and guidance. Next, I would like to thank our beloved HOD Sir, Dr. Mrinmoy Basak, for constantly supporting us in conducting this FDP. Then I would like to thank the first day of the speakers, Dr. Krishna Mohan Chinnala and Dr. Shubham Banerjee, for their excellent presentations and making the FDP an interesting and meaningful. Next, I would like to express my gratitude to Dr. T. Pannirselvam and Dr. Abhishek Kumar Ghosh, sir, second day speakers, for providing their or enlightening the participants with their presentations. Then I would like to thank Dr. Vibhu Prasad Panda, Dr. MD Sarovar Hussain, the third day speakers, for enlightening the participants with their presentations. Then I would like to thank Dr. Kuntam Ravinder, who is the fourth day speaker, for enlightening the participants with his presentation. Then I would like to thank Dr. Tapan Kumar Giri and Professor Dr. Santosh Kumar Mahapatra, the fifth day speakers, for enlightening the participants with their presentations. Then I would like to thank Dr. Supratim Roy, Dr. Kunal Roy, and Chief Guest Dr. P. Chattopadhyay Sir, for sharing their valuable research information and with their presentations. Last but not the least, I would like to thank the organizers, moderators, faculty members, convener. I would like to thank convener, organizers, moderators, faculty members, technical staff, print and electronic media, and participants who had made this FDP program a grand success. Thank you, everyone, for your valuable participation. Thank you. Stay safe and have a nice day ahead. And before this, before my end of this, I would like to hand over this regarding your participation certificates. It will be delivered by Kaushik Narayan, and he will be giving you the instructions. Over to Kaushik.
Okay, thank you, sir. Thanks for your kind words. Now, one announcement for the participants for regarding e-certificate. Those who are actively participated and filled at least 7 feedback form, that is approximately 65%, will be provided the e-certificate in their registered mail IDs within 7 to 15 days. Now, thanks everyone for your presence. Hope this faculty development program is very beneficial for you all. And thank you for having with us, and have a nice day. Thank you.